Hello students, today I am going to apprise you of different biosafety guidelines of Government of India. Basically these guidelines are framed by different government agencies and regulatory bodies to monitor the experimentation on any genetically engineered or any potentially pathogenic microbe or any other animal or plant. As you all know, with the advancement of different techniques and different discoveries, now this is possible to ligate two different DNAs from different origins, and now we are able to clone any gene to insert the genetic material of any other origin in some other host cells. So the implications of this development may cause some hazardous situation. If any potentially dangerous gene or the gene from any pathogen or any genetic material is accidentally releasing from the laboratory by the experimentation, during the experimentation or during the containment, then this may cause a serious hazardous situation for the experimenter, for the society, and for the human beings at large. So Government of India has framed certain guidelines to monitor such experiments in different national and uh, other private institutions, research institutions particularly. These guidelines are known as regulation and guidelines on biosafety of recombinant DNA research and biocontainment 2017. These guidelines are framed by Department of Biotechnology under Ministry of Science and Technology of Government of India. These guidelines basically covers these six domains. One is the storage of such genetically engineered microbes or other pathogenic microbes or other animal cell or plant cell, import of export of these organisms, disposal and emergency situations handling, laboratory uses of these organisms, facility certification or the uh, facility uh, facilities available in the research lab, and also these guidelines are applicable on the manufacture units which are manufacturing any product of GE organism at a large scale. The very first guidelines of Government of India was Environmental Protection Act 1986, which was further released in 1989. And these guidelines were framed by Review Committee on Genetic Manipulation (RCGM), that is controlled by Department of Biotechnology, Ministry of Science and Technology of Government of India. So after that, the specific guidelines for recombinant DNA safety was released in 1990, and these 1990 guidelines were revised in 94 and afterwards in 1998. The latest guidelines which are released recently in 2017 are regulations and guidelines on biosafety of recombinant DNA research and biocontainment, and all are by the virtue of law uh, have the binding to follow. All research institutions and researchers have the legal binding to follow these guidelines in their laboratory, including research institutions, uh, private research laboratories, universities, national institutions, engineering colleges, degree colleges, and all laboratories where any genetically engineered product is being handled or some experimentations are going on, and these are not confined to genetically engineered microorganism only. They have a broad scope to cover, like they cover any pathogenic microbe, any animal cell that is hazardous, any plant cell. So there is a huge scope of these guidelines. So now I will basically explain what are those guidelines, because these are very important. Before starting this course of RDT, you must be aware of the biosafety guidelines so that you may protect yourself and you may protect your lab colleagues whenever you will be working inside the laboratory or later on in your professional life. So, and I would request you all to keep on looking the updated guidelines after the completion of degree also, because this is very important. So basically these guidelines are mainly applicable for manufacture, use, import, export, exchange and storage of hazardous biological material, recombinant nucleic acid molecules and cells, organisms and viruses containing such molecules to ensure an optimal protection of public health and of the environment. So, what is described in these guidelines? This 2017 guideline or any of these guidelines, we are having uh, the uh, following aspects. That is, what is the biosafety requirement? What is the recommendation for laboratory facilities? How the laboratory facility should be designed? What biosafety equipments must be used? What level of personal protective equipment must be worn by the experimenter? What are the good laboratory practices and techniques? And the most important, what is the waste management practices in the laboratory? Because this is the point from where uh, there is the maximum chance of accidental release of any potentially dangerous microorganism from the laboratory. So now I would like to tell you the various objectives of this guideline. So the main objective is to outline or design or frame the principles of containment and establish a minimum standard for laboratory. The laboratories handling genetically engineered organisms, which includes microbes, animals, plant, arthropod, aquatic animals, and any other genetically engineered organism. And another inclusion is non-genetically engineered hazardous microorganisms. These are not genetically modified, but they are hazardous microbes, including parasites, protozoans, algae, fungi, bacteria, virus, prions, and any other potential threat. Another objective is to identify different level of risk. So risk level identification is the another objective. As risk level associated with the GE organism and non-GE organism. Now I would call the two terms GE organism for these organisms and non-GE for this group of microbes. So to identify the risk level. Another is to prescribe the criteria for manufacture, use, import, export, exchange, and storage of any hazardous microbes. And now this is also one objective to publicitize and to monitor that these guidelines are being followed by all other stakeholders involved in the research to ensure the public is well informed about the containment strategies followed in India. So now I'm going to tell you what are different competent authorities of Government of India which monitor, frames the rule and give approval for any experiments related with genetically engineered microbes or non-GE microbes. There are 6 government authorities. First is Recombinant DNA Advisory Committee. This is under Department of Biotechnology, Ministry of Science and Technology, Government of India. This has the role, advisory role. And other than this, there are 3 bodies. One is a Review Committee on Genetic Manipulations. This is under Department of Biotechnology. Genetic Engineering Appraisal Committee. This is under Ministry of Environment and Forest. And Institutional Biosafety Committee. This committee is found in each and every research institutions. This is the mandatory guideline of Department of Biotechnology. that every institution must have one IBSC committee. We also have one IBSC committee in graphic era. So any laboratory that is working on and or that is using any microbe, pathogenic microbe, or if any experiment related to genetic modifications are going on, then you need to have one IBSC committee. So these three bodies have regulatory and approval role. Other than this, there are two more bodies. One is State Biotechnology Coordination Committee (SBCC) and another is District Level Committee (DLC). They have their monitoring role at their level, at state level and at district level. Here the detailed role and the authorities of these competent bodies are written, and for example RDAC under Department of Biotechnology, and its role is mentioned here. Similarly, the RCGM, that is the Review Committee on Genetic Manipulations, this is also under Department of Biotechnology. This is also approval and regulatory body, and this includes the representative from Department of Biotechnology, Indian Council of Medical Research, Indian Council of Agriculture Research, Council of Scientific and Industrial Research, that is CSIR, and other experts in their individual capacity. So these are two national bodies. Other than that, there is IBST committee, Institutional Biosafety Committee. This committee is constituted by all institutions handling hazardous microorganisms and or GE organisms. This committee has one head of the institution, scientists engaged in the RDT work, a medical doctor, and a nominee of Department of Biotechnology. I would explain this in more detail because, as I told you, our university also has a IBSC committee for which we conduct a meeting annually in the presence of one DVT nominee, all experts, where we put all the proposals. As I told you, we also have one IBSC committee in graphic era. So this is the committee. This is the letter from Department of Biotechnology for graphic era university for making the IBSC of graphic era university. So this IBSC committee is uh, renewed in every 3 years, and the DVT nominee is also changed after 3 years. So from 2017 to 20, uh, this committee was valid. I'm also one expert in IBSC committee. So this committee has one chairman, that is the vice chancellor of the university, one DBT nominee that is selected by Department of Biotechnology, Government of India. Like we have Professor Tanuj Kumar Ambani from uh, GB Pant Agriculture University, Pantnagar. There is one member secretary, Pankaj Gautam is the member secretary, and there are some outside experts, one biosafety officer, that is a Dr. Vyas was the earlier medical officer with us, and there are some internal experts like Dr. Pramila, Dr. Nishant Rai, Naveen Kumar. So every year we conduct at least once in a year a meeting where all the proposals of the research scholars or the faculty research which are involving any genetic modification related work are put forward, and this committee gives the approval. This committee checks the facility available. Like we cannot handle the coronavirus in our laboratory because that virus requires BSL 3 level facility. We have only BSL 1 level facility. So if I want to work on corona, then I need to take permission from IBSC. Then IBSC will reject my proposal after visiting my laboratory, and they will give me some suggestion and recommendation to upgrade the facility, biosafety facility, to BSL 3 if you want to handle coronavirus or any pathogen like Salmonella typhi or any human pathogenic virus and bacteria. So this is the committee at institutional level in all institutions of India, which monitors directly, which observe directly the progress, compliances, and regulations related to GE organism or non-GE organism. So here three more bodies which I have described or discussed earlier. GEAC, that is under the Ministry of Environment, Forest and Climate Change, MOEF and CC. And basically, I just wanted to emphasize here, all the GM crops are released finally through this body. Then state government also uh, keep monitoring any experiments on GE and non-GE and the release of such crops or organisms uh, through SVCC. So there is also one more committee at each district level, DLC. So district collector is authorized to monitor the safety regulations in installations engaged in the use of GE or hazardous microorganism at each district level. Containment is very important for GE and non-GE organisms. And the principle behind is to protect all stakeholders from the risk posed by organisms during use of such organism in the laboratory. So this includes combination of facilities. The facility must be in compliances with the guidelines, practices. Experimenter and the lab staff must adapt good practice and procedures to minimize or to avoid the risk of being infected by GE or non-GE organism, and to avoid the accidental release of such potentially hazardous organism from the lab during its disposal. So that is very important. Uh, and this is basically to ensure safety to laboratory workers and outside people and the environment from hazardous microorganisms. So two main aspects are reducing the exposure. Suppose you are doing some experiments on some pathogenic bacteria, then your exposure must be reduced. And second aspect is preventing their escape and establishment in natural environment. As I told you, you cannot dispose any pathogenic bacterial culture or any genetically modified bacterial culture in the sink or in the drain, in the uh, normal drain. So there must be some procedures, some methods to contain these hazardous GE or non-GE organisms. Containment includes elements, risk, and biosafety. So the first is identification of elements. There are two elements. One is the primary element. Primary element means the direct experimenter who has the possibility of exposure to the organism. And the secondary elements are the person outside the laboratory and the environment. So first these two elements must be identified. Next is identification of potential risks associated with the organism. So the organism that is being handled must be assessed for its potential risk. It involves assessment of risk like pathogenicity of the organism for human, animal or plants, what is the mode of transmission and what is the host range of the organism, availability of effective preventive treatment or curative medicine, capability to cause epidemics. Now I would like to exemplify with the COVID-19. So the virus is highly pathogenic and that is pathogenic for human and animals. Mode of transmission is by droplet. So this is very hazardous. Air droplets, and now the evidences are coming for being transmitted through air. Then the ability of effective preventive treatment. So there is no drug or curative or any preventive measure so far against COVID-19. So that is potentially hazardous and very toxic and having a lot of risk. Then capability to cause epidemics. Yes, COVID-19 or CoV, SARS-CoV-2 is capable of causing epidemic. So here are different risk groups. Risk group 1. This is a microorganism that is unlikely to cause human, animal or plant disease. So this is the organism with the least risk. For example, you handle E. coli laboratory strains JM101 or DH5 alpha in the laboratory. So that is safe because E. coli is already present in our gut flora, and this is not toxic to us normally in normal circumstances if your body is in a healthy state. There may be some exceptions. So you may easily perform experiment on E. coli, you may perform experiment on yeast, Saccharomyces cerevisiae, that is safe. So these organisms involve RG, these are RG1, these are under RG1. RG2, a microorganism that can cause disease in human, animal, plant, but the laboratory exposure may or may not cause serious infection to individual, and risk of spread of infection is limited. So these are second group risk. RG3 is a microorganism that usually causes serious or lethal human, animal, plant disease, but does not ordinarily spread from one infected individual to another. Means this is not contagious. Such microorganisms fall under the category of RG3, which are, are potential pathogens for human, plant, or animal, but normally they cannot spread from people to people or from one to another. So, but in RG4, a microorganism that usually causes serious lethal human, animal, plant disease and can be readily transmitted from one individual to another directly or indirectly, like SARS-CoV-2. So the SARS-CoV-2 or any other like HIV virus and many toxic viruses are there, they fall under the category of RG4. Now the third aspect of containment is to determine appropriate biosafety level or biosafety facility for safe handling of organisms. So here are different facilities designated, their safety levels and the type of the organism. Like for the microbe, there are biosafety levels BSL1, BSL2, BSL3 and BSL4. For the animals, the separate guidelines are there, and these are governed by animal biosafety level guidelines, and they are designated as ABSL1 to ABSL4 depending upon different safety levels. Similarly, the plant biosafety levels are different and designated as PBSL1 to PBSL4. Similarly for arthropods and aquatic organisms. So there are different biosafety levels: animal biosafety level, plant biosafety level, insect biosafety level, aquatic organism biosafety levels. Here I would like to uh, apprise you of different techniques involved in the disposal of solid waste. In the next slide, that is for liquid waste, and in the next slide, that is for some sharp articles or objects. So what should be the disposal container and what are the requirement of disposal of different laboratory waste are mentioned here. So kindly go through these requirements and assimilate it in your skills so that you cannot be exposed to, you and the outside world would not be exposed to any hazardous microbe. I would like to tell you that solid waste must be autoclaved, and the autoclaved bag must be marked with a sticker autoclaved or decontaminated. Then autoclave bag must be put into an opaque bag of a definite color, and this must be sealed. And then the bag that is decontaminated and sealed must be stored to be collected by cleaning personnel. And there are certain guidelines for disposal by pollution control board also. Like what we do in the laboratory, we have the BSL-1 facility, and we all the solid waste of the laboratory, for example agar media is there, in which E. coli or yeast cells are grown. So we collect the media in a autoclavable bag used for disposal. Then we autoclave all the stuff, and then give it to the we send it to the uh, disposal facility at cantonment board. For the liquid waste that you are mostly concerned, because any media, any culture media containing cells, liquids coming from petri dishes, culture vials, lab equipments, recombinant nucleic acid, etc., these are covered under this category. So liquid waste must be separated from solid waste, and liquid waste must be decontaminated by using any disinfectant or bleach first. Then after this, flush the disinfected material down the sink, allowing the cold water to run for a period of time, at least 5 minutes. Do not flush non-aqueous solutions such as liquefied agarose or unfiltered broth down the drain, as they will clog and drain the pipes. Liquid waste generated from, this is very important point, liquid waste generated from higher containment laboratory BSL-2 and above should be autoclaved. If liquid waste is coming from BSL-2, then this must be autoclaved before its disposal. Here are the guidelines for sharp object and for pathological objects. Pathological objects must be treated as the solid waste, and they must be incinerated. There are incinerators in the industries or in different research institutions which at very high temperature and in the absence of oxygen, they convert the things to S. So all the pathogenic or the medical waste must be incinerated. So now I'm telling you what are different guidelines, biosafety guidelines, that is the third aspect of containment. So starting with the BSL-1, biosafety level 1, this applies to isolation, cultivation and storage of risk group 1 only, and experiments using RG1 microorganism. There is a specific category called category 1 genetic engineering experiments on microorganisms. So all these category 1 genetic engineering experiments must be performed in BSL-1 facility, and this category includes the experiment which generally do not pose significant risk, because risk group 1 microbes or pathogen, any cell is not very threatful for experimenter or to laboratory workers or community. So these experiments includes insertions of gene into RG1 microorganism, experiments involving host vector system, provided that the donor DNA is originated from RG1 microorganism. This means if any DNA is coming from RG1 microorganism, that is that won't be hazardous for experimenter or the persons outside. Experiments involving the fusion of mammalian cells which generate a non-viable organism. like hybridoma. Any experiments involving microorganisms belonging to RG1, like self-cloning, fusion of protoplast between non-pathogenic RG organisms, or any experiment that you will perform in the laboratory or the students used to perform in our BSL1 uh, facility. But what are the regulations of this BSL1 facility? So before commencement of category 1 GE experiment, the investigators should intimate the IBSC, as we do. We inform the IBSC committee every year for your program to run ki we are running omics technology, we are running recombinant DNA technology, where we would be performing experiments upon E. coli or on the Saccharomyces cerevisiae. We take the approval every year कि we will perform these experiments in the laboratory, and these are category 1 G experiments which are allowed for the students, UG and PG students. Biosafety level 2 regulations are applicable on risk group 2 microorganisms and microbes isolated from environmental samples like soil, water, which are not, you never know what is being grown, all are uncharacterized microbes in that case. So these microbes must be experimented in BSL2 only. And there is one more category 2 genetic engineering experiments. This is a term that is applicable for various points. So these experiments may pose low level risk to laboratory workers, community or environment. As in the category 1 genetic engineering experiment, there was no risk, but here a low level risk is expected. So what are those the experiments? Experiments using any infectious or defective RG2 virus, experiments using non-approved host or vector system, which although that doesn't cause disease in plant, human or animal, but the if the vector and host is non-approved or non-characterized, then you need to perform experiment in BSL2, or if that may be able to cause disease also. And if you are using any system that is replication defective virus, because this may cause the situations like oncogenesis, because in the case of cancer also there is uncontrolled growth. If any vector is replication defective, then it would be very difficult for you to stop the replication of that virus or host or any vector that may be toxic later on or hazardous. And there is one more category, experiments with approved host vector system, but the gene is from a pathogen, means gene is pathogenic, known for pathogenic, or not fully characterized. So not fully characterized means that gene may be pathogenic also. So such experiments must be performed in BSL2. BSL2 is also applicable for the experiments if you are going to delete any gene or transient disruption of expression of gene using any method like antisense technology. You are transiently disrupting the gene expression. So and the implication of this disruption may be pathogenicity, maybe toxicity or survival or fitness. So if anything is falling in this category, then this experiment must also be done in the BSL2 facility. And also if the work is involving fragment of TSE, transmissible spongiform encephalopathy proteins. These are the proteins which may affect the brain. So these must be performed, these experiments with these proteins must be performed in BSL2 facility. And if there is any experiment in which the DNA from RG2 or RG3 organism are transferred into a non-pathogenic prokaryote or lower eukaryote, and the regulation of BSL2 is also executed by IBSC committee. Now what is biosafety level 3, that is BSL3? So if you are dealing with the RG3 microorganism, then you need to follow the compliances for biosafety level 3. And here also, if you are handling environmental samples collected from environment that is likely to contain pathogen, in the BSL2 we were not discussing that the site might contain pathogen, but here if the environmental site from where you are collecting some sample for the isolation of microbe is expected to contain pathogen and expected to cause disease, then such experiments must be performed in BSL3. And another is the experiments on RG3 microorganism or isolates from environment mentioned above, provided that the experiments will not increase environmental fitness and virulence of the microorganism. Now another term is coming, category 3 and above genetic experiment on microorganism. If your experiment falls in this category 3, now what is this category 3? This kind of first of all, this kind of experiments pose moderate to high risk. So they are risky experiments and must be performed following all the guidelines in the facility developed at a level of biosafety level 3. They may be moderate to high risk, causing for laboratory workers, community and environment. Some examples are there. If you are working on the experiments on RG2 and RG3 microorganisms, where insertion of a gene directly involved in production of toxin or allergen or antimicrobial compound. And this is also applicable for insertion of gene into RG3 microorganism. BSL is also applicable in the experiments where insertion of nucleic acids, deletion or rearrangement of nucleic acids are involved, which may result in additional adverse effect on human or animal health or on managed or natural ecosystem. So this may result in the production of certain toxins. So this may affect the ecosystem or the health of human beings and different animals. And if there are certain research involving the introduction of nucleic acid into RG3 microorganism or organism listed in SCOMAT item, you please go through what are SCOMAT item on Google. This is the link given. Another category of where this BSL3 is applicable, genetic engineering of organism isolated from environment where there are reported cases of disease prevalence and possibility of presence of infectious microorganisms. If you are performing such experiments where you are isolating some organism and performing genetic engineering on those organisms from where the cases of earlier prevalence of diseases are reported, then you need to follow the compliances of BSL3. And what are the regulations for BSL3? All category 3 and above GE experiments require prior authorization from IBSC and from RCGM, Regulatory Committee on Genetic Modifications. As I told you earlier, you need to take the permission from government body also other than IBSC. Biosafety level 4 is the maximum containment laboratory, which requires strict training, strict restricted access and the supervision to perform the experiments. And these experiments include experimentation upon RG4 microorganism, its isolation and cultivation storage, handling of samples collected from a patient or environment that are likely infected with the RG4 organism, category 3 and above genetic engineering experiments on microorganisms involving introduction of nucleic acid into RG4 microorganisms or exotic agents. So if you want to work upon uh, potentially pathogenic microbes, you want to work upon their genes and you want to characterize or clone their genes, then you need to work in the facility of biosafety level 4. There are certain things which are not covered under BSL criteria. or compliances. For example, permanently housing, keeping, rearing of any animal, arthropod or aquatic organism for longer than the time required to complete laboratory procedure, or for growing of any plant except those in tissue culture bottles or fully contained in a plant growth chamber. Genetic engineering experiments not covered under any of the above four categories. There might have been many exclusions. So such exclusions will require case-by-case evaluation of the for selection of appropriate containment strategy, and a prior approval or permission from IBSC and or from the RCGM shall be required to initiate experiments. There are certain examples where no BSL facility is applicable and case by case permission is required from RCGM after the evaluation of safety concerns, associated safety concerns of the experimenter and the outside persons and the environment, and also keeping in view the hazardous potential of the organism being handled. All existing BSL 3 and 4 facility must be certified by RCGM. The new BSL 3 and 4 facility shall require certification at the time of commissioning operations as per the format. I will share the format tomorrow in my next class. So the thing here is, you need to take the approval from government bodies and regulatory authorities for establishing a BSL 3 and 4 facility. As you must be hearing and reading in the newspaper also, that coronavirus can be handled in BSL 3 at least. For the testing you need BSL 3 facility, and for experimenting upon the for the research purpose on corona SARS-CoV-2 you need to have BSL 4 facility. Thank you for today. And the second part of biosafety I would discuss in my next lecture, where I would cover the operational guidelines and design of the different biosafety level facilities. Thank you.